So we're going to be going through. So I sent you guys two papers. I gave you a biology one and a chemistry one. So we're going to do 1 hour of biology. And yeah, what I'm going to be doing is I'm going to be going through the questions, and as well as that, we're going to be looking at notes. So hopefully you guys can learn stuff as we do these questions if you're not sure of any of the topics. But yeah, do the questions with me. I don't know if you guys have already done it or you're going to do it while I go through them. But yeah, let me know. Be interactive. Let me know any answers that you guys think you know. Don't worry if you're wrong. So I sent you guys, most of you, I sent you a paper by email. But from next week, I'm going to be just putting them on my website. It's flashrevisionlab.co.uk. Is not only am I going to have the papers on it, I'm also going to have notes, questions, and videos on it. I'll show you that today as we do the questions. It's really useful for revision. So hopefully it'll help you guys. So yeah, any questions, let me know. Just add it to the chat. And let's get started then. So biology paper 1. First question, draw one line from each cell structure to the type of cell where the structure is found. So let me show you guys on the website. You've got, so this is the website. You've got notes. So I'm going to go to the biology section. Before that, let me show you. So on the timetable here, you can download whichever paper that we're going to learn in the next live. So next week's one's going to be this one on the 14th. So you can literally click on it and download it before the live. But yeah, let me show you the biology notes. So I'm just going to go on biology now. And this first question, so nucleus, what type of cell do you find a nucleus in? What do you guys think? Let me know. I'll show you. So we've got two types of cells. We've got prokaryotic and eukaryotic cells. And eukaryotic cells are the ones that have a nucleus. So the answer for that would be eukaryotic cells. So that's the answer for the first one. Which type of cells have permanent vacuoles? So you can go down to the plant cell section and you can see that plant cells have a permanent vacuole there. You can see it over there. So the answer for that would be plant cells only. Yep, well done, Nav. Then we've got plasmid. What type has got a plasmid? It's going to be the prokaryotic cells. That's the only one that's left. Let me close that. Prokaryotic cells we've got here. Structures of prokaryotic cells. They're basically bacteria cells. And yeah, they've got singular strands of DNA. They don't have a nucleus. Eukaryotic cells do have a nucleus. Prokaryotic cells do not. Very blurry for you. Try and refresh. Is there anyone else got a blurry stream? Let me know. Or try and leave and come back if it's blurry. Sometimes the sound goes out of sync as well. If the sound goes out of sync, so if I'm writing something and the sound is like a few seconds later, then just try leave and come back. It works. So yeah, that's the first question. Next up, we've got a plant cell here. What are the names of structures A, B, and C? All good? Okay, nice. So plant cells, here's a diagram of plant cells from the notes that I was talking about. So A is pointing in the middle here. That's a vacuole, permanent vacuole. You can see that in the diagram here on the notes. Then B, okay, so B people get confused because people don't know whether it's a ribosome or a mitochondria. Generally, ribosomes are circularly shaped in diagrams, and mitochondria are usually like this oval shape. So B is a mitochondria. Then C, that's the very outside. The very outside, if we look at this diagram here, the very outside is the cell wall. The inside bit is the cell membrane. So the cell membrane would be this bit here. The outer part would be the cell wall. So yeah, well done. Cell wall would be the answer for that. So the answer is vacuole, mitochondria, and cell wall, which isn't there. Vacuole, no, why did I write mitochondria? Sorry, not mitochondria. This is a ribosome, isn't it? I said the circular ones are ribosomes. Sorry. So that is a ribosome. So the answer is this one. Vacuole, ribosome, cell wall. Okay, next question. Yes, thank you, Georgie. A student observed slides of an onion cell using a microscope. Figure 2 shows 2 of the slides the student observed. Describe how the student should adjust the microscope to see the cells on slide A more clearly. Okay, so what we can do, go on the website. Let's go on microscopes. Let's look at the parts of a microscope here. All right, so we've got eyepiece, we've got the adjustment knobs, fine adjustment knobs, and coarse adjustment knobs. So what you want to do is you need to try and focus. So you can see here it's out of focus. In slide B, it's in focus. So how can you go from out of focus to in focus? You just need to turn the fine adjustment knob. To focus it. There's only 1 mark, so that's enough for 1 mark. Describe how the student should adjust the microscope to see the cells on slide B more clearly. So you can see here, you can't really see the cells. The cells are quite small. So what you want to do is you want to try and zoom in. So how could you zoom in a microscope? What could you do to zoom in? So that's when Ribosome, do you need to mention any direction? No, you don't need to mention the direction. You can just say adjust the knob. So that's fine. Describe how the student should adjust the microscope to see the cells. So basically, you've got the objective lenses here. So you can see here, there's like 3 different lenses. You want to adjust it to get the higher powered lens. So all you have to do is say that you rotate the objective lenses. That's 1 mark. Second mark, to a higher power. And that should be enough to get the 2 marks. Next question. A student made necessary adjustments to get a clear image. Okay, so we got the cell here. The real length of the cell was 280 micrometers. Calculate the magnification. So for this one, we need the equation for magnification. The equation for magnification is size of the image over size of the real object, the actual object. So magnification is equal to size of image over size of actual object. So the image is what we're seeing here. That's the image. The real length, that's the actual length of it. So what you want to do is image over actual length. But what do you need to remember? You need to remember that this is in millimeters. This is in micrometers. Do you guys know how to convert from millimeters to micrometers? What could you do? So basically, I'll tell you on the side here, 1000 micrometers is equal to 1 millimeters. So what we would need to do is we'd need to convert one into the other. So let's say we want to convert the 112 millimeters into micrometers. we just want to times that by 1,000. That gives you 112,000 micrometers. Now we can put that into this equation here. And that gives you 112,000 divided by 280, because they're both in the same units. They both have to be the same units for you to work that out. And then you put that in the calculator and you get an answer of not quite 1,000. Anyone else got another answer? So you do 112,000 divided by 280. Put that in the calculator. You should get 400. Yeah, well done, Sarah. Times 400. So magnification there is 400. Good. So I was just doing the conversion. Oh, okay, okay. Yeah. The conversion, yeah, you times it by 1,000. And then, yeah, the final answer is 400. So I hope that makes sense. Guys, remember, just let me know. If something doesn't make sense, let me know. I can explain it again. On to the next one. So these papers, generally the questions at the start of these papers are going to be quicker. And then the ones at the end are usually the longer questions. Figure 1 shows one type of white blood cells. What's structure A? Hopefully that's straightforward. You can just use this diagram here. You can see the outside bit is the cell membrane. Answer cell membrane for that one. White blood cells help to defend the body against pathogens. How do white blood cells do this? You got to look carefully. It says 3 marks. It says tick 3 boxes. Anyone know what the answers are? How do white blood cells fight against pathogens? I'll get the notes up while you guys tell me the answer. If you know the answer, there's 3 boxes you need to tick. So on my website, let me go on infection and response, communicable diseases, and here, the immune system. So we've got engulf pathogens. Yep, well done. That's one of them. Produce antibodies. Yes, and produce antitoxins. Well done. So here you can see here the immune system, phagocytosis. That's when the white blood cells engulf and digest the pathogens. Antibody production and antitoxin production. So those are the 3 answers. That's how to get 3 marks for that one. On to the next one. So this question is all about measles. A person can die from measles. Figure 2 shows the number of cases of measles in England and Wales between 2012 and 2015. Use figure 2 to calculate the decrease in the number of cases between 2012 and 2015. Okay, so 2012 and 2015. What you need to do first is see what numbers there are. What do you guys think? What number is this? That's 2012. 2015, that's on the second square, so that's going to be down here. What do you guys think those 2 numbers are from looking at that? This one and this one. So each square, so you've got 10 squares going to 500. So that means each square is 50, isn't it? We've got 2 squares here. So that bottom one is going to be 100. And then the top one is 2000. There's 1 square above 2000. So that's going to be 2050. Those are the 2 values that we've got there. So you want to work out the change. So you just want to minus the 2. 2050 minus 100. Work that out. That will give you 1950. That's the answer for that one. I hope that makes sense. So just one reason for the decrease in the number of cases of measles between 2012 and 2015. What could those reasons be? Mainly because probably people were vaccinated. Just like COVID. In COVID, there was loads of cases in 2020, 2021. And now there's not as many cases. There was a huge jump because people were vaccinated. So you can literally just say that. Okay. Antibiotics cannot be used to treat measles. Why is that? Let me go on to some types of communicable diseases. Measles is a virus. Yes, well done, Georgie. Measles is a virus. Antibiotics can only kill bacteria. It cannot kill viruses. So that's the answer. How many marks is it? 1 mark. Yeah, you can just say measles is caused by a virus. Good. Then we got gonorrhea. Is a disease caused by bacteria. So this one can be treated by antibiotics because it's caused by bacteria. One way to control the spread of gonorrhea. So it's a sexually transmitted disease. So you could say use a condom, or you could say vaccination again. Vaccination, that should be fine as well. Either one are acceptable. Okay, next one. Scientists investigate how effective different antibiotics were at killing gonorrhea bacteria. So what have they done? They've grown gonorrhea bacteria on agar in a petri dish, place 1 paper disc soaked in water. So that's this thing here. So that's soaked in water. Then the other 4 are soaked in different antibiotics. So we got A, B, C, and D here. They're all soaked in different antibiotics. All of them use the same sized paper discs, same concentration of antibiotics. So what's happening here? So all this gray part in the circle basically represents the bacteria. So that's the bacteria. The white part basically represents no bacteria. Why? Because the bacteria have been killed. So the antibiotic has killed the bacteria. That's what's causing those circles around it. So you can see here, the paper disc soaked in water has no ring around it because the water is not killing the bacteria. One control variable the scientists used. So control variable is just something that you keep constant to make it a fair test. They've literally said it in the question. They've told you. So Abhishek said, I thought when it came to bacterial pathogens, vaccinations weren't allowed as an answer. No, you can say vaccination for this. This was fine. But yeah, the other answer was you could say use a condom as well, because it's a sexually transmitted disease. You could say that as well. Give 1 control variable the scientists used. So it says here, same size paper discs, same concentration of each antibiotic. Either one is enough to get you the mark. So I'm just going to put same sized discs. Suggest why 1 disc was soaked in water. Anyone know why we did that? Remember, that's the one that didn't produce a circle around it. That's this one here. No worries. Could you refer to the amount of bacteria as a control variable as well? Yeah, you can. You can say the concentration, not the amount of bacteria. You can say the amount of antibiotics or the concentration of antibiotic. That's acceptable as well. But you can't say amount of bacteria. Oh, no, actually. No, you wouldn't say amount of bacteria because they're all being put in the same petri dish. So it's not really something that you're it's not a different experiment. If it was like loads of different petri dishes, then you could say that. But for this one, because it's all happening in one, you don't really you can't really say that. But you can say concentration of antibiotic. Okay, so yeah, to control one. So this is a control. Is used as a control. A control is different to a control variable. Don't say a control variable. A control variable is something that you keep constant to make it a fair test. A control is something that you use to compare your results. Because you know that antibiotics are going to kill the bacteria, but we soak it in water just to prove that those bacteria aren't being killed by anything else. They're not being killed by water. So we use that as a control just to show that it is actually the antibiotics that are working and killing the bacteria. So you can say use as control as the answer, or you can say to check that water didn't have an effect. So to show that water did not have an effect. Either one's fine. You got that wrong in a mock test. Okay, hopefully in this one then, in your actual exams, you won't get it wrong. Which antibiotic in figure 3 would be best to treat gonorrhea? So you can see here, what did I say? I said the area which has no bacteria in it is probably the best, the most area with no bacteria in it is probably the best one. That'll be this one here. A, because it's got the biggest area with no bacteria in it. So you'd say antibiotic A. Reason, because it has the largest area with no bacteria in it. Yeah, well done. Okay, next one. So this is a practical, required practical. Let me show you on the website. This comes under osmosis. This is on osmosis practical. So you can search it. You can search the topics. This osmosis, transport in cells. This practical here. So it's the one with the potato cylinders in a liquid. So what's happening here? So they've cut 3 pieces of potato, same length. That's important. Dry each piece with a paper towel, weigh each piece, place each piece in different concentrations of sugar solution. So you can see here we have pure water here, sugar solution with only 0.4 mole per dm cubed, and one with 0.8 mole per dm cubed. So they've got different concentrations. Why did the student dry each piece of potato before weighing it? Why did they dry it? Reason for that is because water is going to add mass to it, isn't it? If the potato is wet, then you're not just measuring the weight or the mass of the potato, you're also measuring the mass of the water. The water on the potato would add to the mass. Get rid of excess. Yeah, I don't think that's enough to get the mark though. Get rid of excess so that it doesn't get counted when you're weighing mass. So it's close, but I don't think you'll get marked for that. Next question. What 2 changes would you expect in potato in tube A after 2 hours? So that's the one with pure water in it. What do you think will happen to this piece of potato? So here, we've got the information here. So it's all to do with water concentration. It's pure water, so it's going to increase in mass. Yeah, well done. It's going to increase in mass. The reason why is because outside here, we've got a high water concentration. And inside the potato, in the cells, we have a low water concentration. And that's the definition of osmosis, isn't it? So if you look at our notes here, osmosis is a process where water moves across a partially permeable membrane from an area of high water concentration to an area of low water concentration. So it's high water concentration because this is literally pure water. You can't get higher than pure water. Potato has a low water concentration. So what would you expect to happen after 2 hours? You'd expect it to increase in mass because the water is coming into the potato. That's one of the answers, though. It says tick 2 boxes. What's the other one? So loads of people would have missed that out. They would have just ticked one box there. So it increases in mass also. Doesn't break into pieces. No, doesn't decrease in hardness. No. It's going to increase in length as well. Increase in mass, they get bigger. So you can see here in the notes as well. The length gets longer and the mass gets heavier. Yeah. Good. Part C of the question, water moves into and out of the cell by the process called, so we've just talked about this already, it's called osmosis. Water would move into the potato cell in tube A. We've already mentioned that. That's why there's an increase in mass and increase in length. The solution outside the potato in tube A is at a something concentration. So it's a higher water concentration, but it's not saying water concentration, it's saying concentration. So you would say lower concentration. Does that make sense? Because if it's a higher concentration, it's a higher water concentration, but a higher water concentration basically means it's more dilute. If it's more dilute, it means it's a lower concentration. How do you know in the question that they're not referring to water concentration? Because, that's a good question actually, because they haven't said it. If they say water concentration, then you'd have to say a higher water concentration, but they just said concentration. A high water concentration means a low concentration because it's dilute. Yeah, they would tell you. They'll tell you if it's water concentration, then you have to say higher. But if it's just concentration, they mean the amount of sugar. The amount of sugar in it. So that would be that would be lower because there's more water in it. Hope that made sense. Tell me if it didn't. I'll explain it again. Is potato for water? What do you mean, sorry? Oh, what do you mean by that? Sorry, is potato a water? So the potato is has water in it, but it's also got sugar in it. So if this was like a cylinder of potato, inside of it you have a sugar solution. You have a sugar solution in there, but it's we don't know how concentrated that solution is. Oh, it's potato A in water. Yeah, potato A is in water. Look, it says water, and it says the sugar solution is 0.0 mole per dm cube. So it's pure water. Does that answer your question? The potato in tube B did not change. Give one conclusion that can be made. Okay, so, okay, good. Tube B, it says that the potato did not change size. Why could that be? So let me show you in the notes here. So there's 3 scenarios you can have in this practical. You could get the cylinders gaining mass, getting bigger. So that was A in this example. They could get smaller. That's probably this one. It says here that in tube B it stays the same. If cylinders in sugar solution stay the same, the water hasn't moved in or moved out of the cell. That's because they have the same water concentration as inside the potato. So remember I said here, there's a sugar solution inside the potato. That solution has the same concentration as outside. So the solution inside the potato is the same as the liquid outside. So that's the, yep, well done, Georgie. The concentration of water in the potato and the surrounding solution is the same. Yeah, that's the answer. So you can say the concentration you can say the same, or we know what the concentration is, isn't it? It says in the question, it says 0.4 mole per dm cubed. So you can say it's 0.4 mole per dm cubed, or you can say it's the same as the solution. Both are acceptable. Okay, moving on to the next question. Figure 2 shows the root of a germinating seed. 2 ways a root is adapted to absorb water efficiently. So you can see here there's loads of root hairs sticking out. So what that does is it increases the surface area. That comes under specialized cells. Another adaptation is in the root hair cells, you've got thin walls. The thin walls means that water can diffuse across osmosis more efficiently. So walls of cells are thin. Okay, let's check how many questions we've got left. About halfway mark. Right, so we've got some longer questions coming now after these multiple choice questions. What is the correct balanced equation for photosynthesis? Photosynthesis is probably the most important topic in biology. What do you guys think? It can come up in any paper. So I'll tell you which topic it comes under on the website. Bioenergetics, photosynthesis, the reaction of photosynthesis. We've got it right here. So no, it's not the first one. It is, yeah, this one. So this one here, people always get this confused. This is something else. This is aerobic respiration. Aerobic respiration is the opposite of photosynthesis. Photosynthesis is that. Carbon dioxide plus water forms glucose plus oxygen. So we got it here in the notes. Yeah, exactly. So plants give off oxygen. So that should be in the products. They take in carbon dioxide. They take in water. So from the soil, they take in water. From the air, they take in carbon dioxide. They produce glucose. That's their food. And the byproduct is oxygen. So the answer is 3. What type of reaction is photosynthesis? Not aerobic, not oxidation, either endothermic or exothermic. So endothermic means it takes in energy. This is chemistry, but you do need to know this for biology. Exothermic means it gives out. Yeah, it's endothermic. It's taking in energy. Where does that energy all come from? It comes from the sun. So remember, in photosynthesis, the plants absorb sunlight through the chloroplasts. So they use that energy from the sunlight to produce glucose. So because aerobic respiration is the opposite of photosynthesis, you would say that aerobic respiration is exothermic. Okay, right. We've got another experiment here. Student investigated the effect of light intensity on the rate of photosynthesis. So that's one of the factors that affect photosynthesis here. Rate of photosynthesis, light intensity, CO2 concentration, and temperature. Those are the three factors. This one is investigating light intensity. Sodium hydrogen carbonate solution releases carbon dioxide. Okay, so let's remember that for the pondweed. So what's happening here is you've got the LED light source here producing light. That's going to the pondweed. That's a plant, so it's got chloroplasts in it, so that photosynthesizes. So the method, place the pondweed at 5 centimeters from the light source. So that's what they've done here. Measure the rate of photosynthesis by counting the number of bubbles. So the bubbles are basically oxygen. Remember, that produces oxygen. Photosynthesis produces oxygen. And they counted that for 1 minute. Repeat the pondweed at 10 centimeters, 20 centimeters. Counting the number of bubbles produced in 1 minute is not an accurate way to measure the rate of photosynthesis. So just 2 ways that the method could be improved. Any ideas? How could we improve this method rather than counting bubbles? Because that's not going to be very effective. The bubbles are going to come out very small. It's going to be hard to spot them, and they're going to move around quite a lot. Any ideas? Collecting the gas. Yes. Do you know how you can collect the gas? What can you use to collect the gas? We come across this in chemistry. Using a gas syringe. But I think saying collecting the gas is fine. You get the marks for saying collecting the gas. Actually, no, you wouldn't get the mark for that. Because you have to be able to measure it. You have to measure the volume of gas. So collect it and measure it is what you need to say. Collecting gas using a gas syringe, that'll get you a mark. Anything else you can say? Another thing. So in questions like this, guys, you should try and read the question. They try to give you clues in the question, especially when they've got loads of writing here. They've said here, they've measured for 1 minute. That's probably not long enough. To make it more accurate, you can measure for longer. Measure the volume of gas for longer. So increase the time. That's enough to get the second mark. Weighing the solution. No, that's not going to be very accurate because gas bubbles are very small. So when you weigh it, if you lose like 1 extra bubble, it's not going to change the mass that much. So that's not very accurate. That's not an accurate way of measuring it. But good idea. All right, on to the next one. The LED light source does not get hot. So normal lamps do get hot, but LED does not. Why is that important? Any ideas why? Why is it important that the pondweed remains at the constant temperature? What could that be? something to do with what's on the notes right now. Exactly. Well done. It doesn't speed it up. Look at this. Look at the factors that affect photosynthesis. Temperature. Yeah, good. So temperature increases the rate of photosynthesis. So what you want to do is you want to keep temperature constant so it doesn't increase the rate. So to get the 2 marks here, so 1 mark is to say temperature affects the rate of photosynthesis. What else can happen if it gets too hot? So that's only 1 mark. The second mark is actually very difficult to get. What can happen if the temperature gets too hot? So I'll show you the graph for temperature, this one. What happens here if the temperature gets too high? Why does that temperature go down low when the temperature gets too high? Here, it damages. Don't say damages. It denatures. So it denatures the enzymes in the plant if the temperature gets too high. So that decreases the rate of reaction. So to get second mark, you could say high temperature can cause enzymes to denature. Okay, right. Next one, this is a tricky one. Light intensity can be calculated using the inverse square law. So what does this mean? It means light intensity's I is proportional to 1 over the distance squared. So we're expected to use this relationship to explain how light intensity changes as the distance is doubled. Okay, so you must include calculations. They've given us numbers here, 5, 10, and 20. These are all the distances. So in this equation, that's this. So what you need to do is you need to put 5, 10, and 20 into this relationship. So if we put 5 in it, intensity is equal to 1 over 5 squared. 5 squared is 25. So you basically got 1 over 25. Now we're going to double this and get 10 because the question says 10 centimeters. We can put that into the relationship again. 1 over 10 squared. 10 squared is 100. So that gives you 1 over 100. Then we've got 20 centimeters. So we're doubling it again. Times 2 times 2. Intensity is 1 over 20 squared. Twenty squared is 400. Okay, I hope everyone's following. Tell me if you're not. Tell me if it doesn't make sense. So what I've simply done is I've subbed in the values they've given us into the relationship that they've asked us to prove. Okay. And the question's asking, how does light intensity change as the distance changes? So the distance is this. The distance is doubling here. Distance is doubling here. This is what the light intensity is. So what's happening to the light intensity as you double the distance? So for the first one, what's happening? How do you get from 1 over 25 to 1 over 100? What do you guys think? 1 over 25 to 1 over 100. It's a tricky one. I think I got some notes on this as well. Let me look for it while you guys try and figure that out. So that comes under photosynthesis. I think it's the practical. Yeah, we've got it here. Exactly. Well done. Well done, Rianch. So what's happening here, to get from 1/25 to 1/100, what you're doing is you're making it 4 times smaller. 4 times smaller. So the question asks, explain how light intensity changes as distance of pondweed from the light source is doubled. You would say light intensity is quartered. If you're dividing it by 4, that basically means you're quartering the number. You'd say light intensity is quartered. But yeah, that is, I'd say that's a quite difficult question for GCSEs. Let me know if you want me to explain that again. Next question. A student's results are shown in the table below. Predict how many bubbles would be produced in 1 minute if the pondweed was 40 centimeters. Okay, so what have we just learned? We've learned as you double the distance, this quarters. Roughly quarters, okay? Not exactly, because it's not like 100% accurate. So if you want to test 40 centimeters, what do you think is going to happen? So let's look at this bottom row here. We've got 20 centimeters. So when it's 20 centimeters away from the plant, you've got 8 bubbles produced. The question is asking 40 centimeters. To get from 20 to 40, you're basically doubling it. So what do we need to do here? Let's look at what we just proved here. Dividing by 4. Every time you double the distance, the light intensity is quartered. Double the distance, light intensity is quartered. So you've got to do the same here. We doubled the distance. So if that's 8, you've got to do 8 divided by 4. Gives you an answer of 2. So you'd expect to see 2 bubbles. Again, I'd say that is quite a hard question. All right. Any questions on that? So yeah, I've got some few notes on this that you can look at after this session. So yeah, you can look at notes, and then on each section, you can click on questions. So if you click on questions, you can find like loads of different examples of questions on that specific topic, and then you can check your answers. Oh, so this is the one that we've just done here. Is that the same one? No, it's a similar one, but yeah, you can check your answers here. So in each section, you can find notes, questions. You can even check the spec points. So here, this is exactly what the exam board wants you to learn. So if you understand everything in the spec points, then you're pretty much ready for that topic in the exams. But anyway, let me carry on. Got 10 minutes left before we switch to chemistry. Describe how the student could change the method to investigate the effect of carbon dioxide concentration on the rate of photosynthesis. You should include how to change. Is it for combined? So the notes right now is for combined, but everyone in triple needs to learn it. So in triple, you have to learn all the combined, and it's higher as well. But I'm trying to add the triple, the separate content as well. And that's my next aim. But right now, yeah, it's got most of the topics that you need to know. How to change the independent variable. So what you're trying to do is investigate the effect of carbon dioxide concentration on the rate of photosynthesis. So you're basically doing this same experiment, but instead of changing the light intensity, you're trying to change the carbon dioxide concentration. How can you change carbon dioxide concentration in this experiment? They have given you the answer in the question if you read carefully. Anyone know? I've circled it. Not circled it, I've underlined it. So you can see here, sodium hydrogen carbonate solution releases carbon dioxide gas for pondweed. So if you want to investigate the carbon dioxide concentration, what you want to do is you want to change the concentration of sodium hydrogen carbonate. If you use more sodium hydrogen carbonate, so right now they're using 0.2%. If you want to increase the amount of carbon dioxide, you use a higher percentage, so maybe like 0.4%. And then you test at 0.6%, and then 0.8%. But what do you need to keep constant? You need to keep the distance constant. So this time, you don't increase the distance. You keep it at 5 centimeters, or you keep it at 20 centimeters. Just make sure it stays the same. So that is, the distance would be the control variable. 2 control variables. We need another control variable. So control variable and independent variable. So I've told you what the independent variable is. So the concentration of sodium hydrogen carbonate and the control variable is, we'll say, the distance from the light. Anything else we can say? Something that we've already mentioned, the temperature. Remember I said that the temperature affects the photosynthesis. You can say the temperature of the solution. Okay, I think we're on the last question. We're nearly done here. Power of light. So power of light, yeah, you can say light intensity. That'll get you the mark as well, because that's the same as distance from the light. You're basically changing the light intensity. That'll still give you the mark as well. That's fine. Okay, next one. Measles is a serious disease. The person can die from measles. The table below shows the number of medically confirmed cases of measles. Okay, so we can see here, there's a big jump here from 2013 to 2014. So just one reason why the actual number of cases of measles in England and Wales might be higher than that is the one that is shown in the table above. So basically saying, why are these numbers probably higher? So why, same with COVID actually, like we got given COVID cases, the number of cases every year. Why are they likely to be higher than the ones that they told us in the news? Yep, thank you, Tina. Because of unreported cases. There's going to be, not everyone's going to report that they have measles, basically. Unreported cases would mean that you don't tell the doctor, basically. So the doctor doesn't know, so then they don't pass it on and record the data. Calculate the percentage decrease. So we had a similar question with the bar chart earlier. This time they've given us numbers and they're asking to calculate the percentage decrease from 2012 to 2015. 2012, 2015. How can we calculate the percentage decrease? This is like a maths question here. What could we do? Any ideas? So first work out how much is decreased by 2030 minus 91. So work out what 2030 minus 91 is first of all. That is 1939. We want the percentage decrease. So we want this as a percentage of the original. The original is 2030. So what you want to do is 1,939 divided by 2030. You want it as a percentage, so you want to times it by 100, and that gives you, yes, thanks Tina, 95.5. Nav said 96. Both are fine. So in these questions, be careful with the number of significant figures that you give your answers to. If they ever tell you, give your answer to 2 significant figures or 3 significant figures, make sure you do it, because that would be worth a mark. If they don't tell you, then roughly use what's given in the data. So see here, it's around 3 significant figures. This one's 2 significant figures, so either one will be acceptable. But if they specifically tell you to use a certain number of significant figures, make sure you use that. Next question. One reason for the decrease in the number of cases of measles is that more children are vaccinated. Vaccinating a large proportion of population reduces the spread of measles virus. Explain why. Reason why is because more people are immune, so they don't become ill, meaning they're less likely to pass it on. It's what we call herd immunity. So more people are immune, so less become ill with measles. So they are less likely to pass it on. All right, on to the last question of bio. So the graph below shows the concentration of measles antibodies in the blood of a boy. Explain the differences between antibody production after the vaccine injection and after exposure. So vaccines, so that would be in the disease section of the website, infection and response. Yeah, vaccination. So what you do in a vaccination is you introduce a dead or inactive pathogen. Okay. So this question, you got to look at the 2 differences that they're talking about. So the vaccine injected and the exposure to measles. So when you're injected with a vaccine, you've got a dead or inactive pathogen within you. So your body is responding to that by producing antibodies. It's the white blood cells that produce the antibodies. But after, so I've got it here. So once all the pathogens of a disease have been killed, most of the lymphocytes that produce the antibodies are no longer needed. So most of them die off. So once you fought off all the measles pathogens, most of them die off, but some of them still remain, and we call those memory cells. Okay, so we got some memory cells. So the memory cells remain so that if you're ever exposed to the measles virus again, the white blood cells will rapidly produce the antibodies. So what's the 2 differences? You can see here, this one's much higher. The line is much steeper. See, look at that line, and then look at this line. So after exposure to the measles virus is much steeper. So that means the antibodies are being produced much faster. So they're being produced at a greater volume, much faster. And the last thing you can talk about here is after. Look how slowly this one decreases. Look how slowly this one decreases compared to this one. This one goes back to 0 very quickly. This one decreases very slowly. So explain the differences. That's what you need to talk about. This is a 6 marker. So it's quite hard to get all 6 marks here. But things you can say is there's a greater number of antibodies produced. That's why this peak is higher. another thing you can say, so I talked about the steepness, antibodies are produced more rapidly. And remember, they're decreasing more slower. You can say that the antibodies remain in the body for longer. Okay, and then we need to explain that. Why is that? It's because of the memory cells. So after the first infection, so I'm talking about this one here, after the first infection, white blood cells remain in the body as memory cells. So when the pathogens reappear, they are rapidly reproduced. Okay, my laptop's about to die. Let me just put the charger on and we'll move on to chemistry.